Now we'll learn what is isotonic solutions, what is hypotonic solutions, and what is hypertonic solutions, and what happens when a cell is placed in such solutions. So for hypotonic, we'll start from the hypotonic solution. Here is the hypotonic solutions. Now hypotonic solution is a dilute solution. Dilute means in a hypotonic solution there is a large amount of water. So this is a cell that is put in a solution that has got a lot of water in it, high concentration of water outside the cell. However, inside the cell, the concentration of water is very less. So now here we have low concentration of water, low concentration of water here inside the cell, but outside there is a high concentration of water, very high concentration of water. So what you expect the water to happen? The water will begin to rush into the cell. See here, the water is rushing into the cell, and because it rushes into the cell begins to swell. Here in a hypotonic solution, which is a dilute solution. Why dilute? There is lots of water, and when the cell is put into it, the water rushes into the cell. And however, we must note that this process of the movement of water is called osmosis. This process is called the osmosis process. Osmosis takes place and water rushes into the cell. Now let's get on to an opposite situation wherein the cell is now put in a hypertonic solution. What do you mean by hypertonic solution? Concentrated solution. Concentrated solution means there is very less water. See, the other molecules are that of a solute, maybe salt or or sugar. Okay, so outside there is less water, but there is lots of other things. Concentrated, maybe concentrated sugar solution, lots of sugar in it, or concentrated salt solution. So these are all sugar or salt, less water and a lot of sugar or salt or other solute. So again, see, there's a lot of this. This could be these molecules, yellow molecules out of the solute. Solute means it could be sugar or it could be salt. Okay, but the fact is outside the water is very less. Okay, so inside the water concentration is very high because outside there is less water. Inside the concentration becomes more than the outside. So have you seen here, inside there is a high concentration of water, outside there is a low concentration of water. So the water will start leaving the cell, and because the water will start leaving the cell will begin now to shrink. So here I've written the water rushes out of the cell and the cell shrinks. The net water rushes out. You know, there is some water that enters, but more in this case, there is more water that rushes out and very little water that enters. A little water may enter, but most of the water is rushing out. And now for the third one called the isotonic or normal solution. And in this have you seen the solute and solvent are equal. The water, the concentration of water and the solute is almost equal. And inside the cell also there is the same amount, same concentration. So inside same. And here the concentration is same. So the amount of water entering the cell is equal to the amount leaving the cell. Here entering the cell equal to the amount leaving the cell. Entering the cell equal amount leaves the cell. So enter same. So there is a net zero flow of water. The net flow of water is zero. Okay, net zero flow of water, and the cell remains the same. There is no increase, remains the same size. There is no increase or decrease in the size. So we'll summarize it again. Here is another picture. See, we have the red blood cells and they are put in different solutions. The one is put in an hypotonic solution. Hypertonic means with lots of water around it. So what happens in this case? The water begins to enter the cell, and because the water enters the cell, you can see in this case it swells. Then in the next case, it is put in a isotonic. Isotonic, the solution, the concentration is the same. The concentration of water inside and outside is the same. So the amount of water leaving is equal to the amount of water entering. So the size of the cell remains the same. Size remains the same. And here we have, it is put in a hypertonic. Hypertonic means very less water. There is less water. However, there is a lot of other solute in it. Maybe there is a lot of salt in it or sugar in it. Fine. So the concentration of water inside the cell is more, while the concentration of water outside the cell is less, and the water moves from the cell outside, begin to rush out, and because of this the cell here shrinks. Okay, so three things happen to these red blood cells. If they're put in hypotonic, they swell. If they're put in isotonic, they remain the same because the concentration is same. If they put in hypertonic, the cell shrinks. Now in this next diagram, we see the same what we have done here. If an animal cell is put in an hypotonic solution, we know now water begins to rush in, and the water keeps rushing in till the cell in the end ruptures. Have you seen here? The cell has ruptured because too much of water is entering into the cell. This membrane is so thin, it cannot withstand this pressure, so it ruptures. If it is kept in the isotonic solution, the amount of water entering is equal to the amount of water leaving, normal the cell. But when it is kept in hypertonic solution, we've seen the water leaves, and because the water leaves, the cell begins to shrink. It becomes smaller because water is leaving the cell. That is for an animal cell. But now in plant cell, have you noticed here? It is hypotonic here. There is no change in shape. Isotonic also, no change in shape. In hypertonic also, no change in shape. Because we have a very thick membrane. The thick membrane is called the cell wall. This cell membrane withstands that water. It does not allow the cell to swell up. We'll learn more of this in detail when we learn plasmolysis, but we will learn more. So the point I'm trying to make here is a plant cell, when it is put in hypotonic, isotonic or hypertonic, then nothing happens. The size remains the same because it has got the cell wall, not cell membrane. It has got a cell wall that protects, that can withstand the pressure. We call the cell wall in the cell membrane inside. This cell membrane inside, just like here, but over and above it has got the cell wall, the thick cell wall outside. This thick cell wall, which does not allow the cell to burst. It protects the cell from bursting. Now we'll see how aquatic plants and animals get oxygen. So here's a picture of aquatic. You can see the fish and aquatic plants there. And there is a lot of oxygen out here in the air. We know in the air we have about 20% oxygen. 20% of our air is oxygen. Now this oxygen slowly enters into the water. It dissolves into the water by the process of diffusion. So this oxygen would be entering here, and this happens also by the process of diffusion. So it is because of diffusion that these aquatic plants and animals get the oxygen that they need. This is all oxygen that they require. Now plants also use osmosis a lot, and they use osmosis to take in the water. So here is the soil, and inside the soil there is water. We'll put it another color. So here is water outside the soil. So outside the soil there is a lot of water. There is a high concentration of water. So what would you expect? Inside there is less water. So what do you expect the water? The water will now begin to enter into the root. And again, this process of water entering into this root is called osmosis. By the process of osmosis, the water enters. Now remember, we'll then learn the difference between osmosis and diffusion. Osmosis is a kind of diffusion, but osmosis is when water moves up and down. Diffusion is the movement of any substance from one place to another, but osmosis is only the movement of water. So plants also use osmosis to take in water. Many unicellular organisms like the amoeba, okay, we have the amoeba, and this amoeba also lives in water, and the amoeba takes in water or oxygen or other substances. It takes in substances, it throws away our substances, all by the process of diffusion. It also extensively uses diffusion for the substances to enter and for the substances to leave.